Hello, my name is Helena, and now I'm presenting the poster Grape Cell Wall Changes Upon Infection by Different Fungal Pathogens. So first of all, the plant cell wall. The plant cell wall is a complex matrix of polysaccharides and glycoproteins, and it is the first layer in the cell wall, and it brings shape and rigidity to the cells in the plants. It is also one of the first lines of defense, which makes it crucial in plant and pathogens interactions. Other first lines of defense were, for example, the cuticle, the wax. So the cell wall is mostly composed by cellulose, which is the most abundant polymer in the cell wall, and it's linear chains of glucose monomers that don't cross and don't branch. It is responsible for the structure. Then we have hemicelluloses. Hemicelluloses connect the cellulose fibers mainly, and they can be branched. They can have side chains with many types of monosaccharides like xylose, glucose, mannose, and that will give them different families. Then we have the pectins. Pectins are responsible for cell-to-cell adhesion and as well for the cell wall hydration. So all the water retention is managed by pectins, and that will also shift its mobility and rigidity. And they have a backbone of galacturonic acid or galacturonic acid alternating with rhamnose, and they can be very complex like rhamnogalacturonan 1 and 2, and they have a lot of side chains usually, except for homogalacturonan. Then we have glycoproteins, which are proteins that are in the cell wall that have a glycopart attached to it, and they intervene in the cell wall by as well as defense, but as well in signaling and connecting and disconnecting cell wall components. So as we can see in the scheme in general, we have our object of study, which are grapes. This is the scheme of a plant cell, and we can see the cell wall here in green, and if we magnify we can see that there's this pattern where we have stripes of cellulose and then we have interlinked everything else. So how does this relate with grape diseases? So grape diseases are quite catastrophic for economics and production of grapes and consequent wines. There's three main diseases that we study here in our lab. The first one is botrytis. So botrytis affects many fruits, not just grapes, and even we can detect it at green stage, which is quite rare to see this phenomenon in the world. So it gives this gray mold disease that kills the cells and does it by secreting enzymes that destroy the cell wall. So in the model, we can see that we have our botrytis that secretes the enzymes that will mainly be pectinases, and they will they will destroy or degrade homogalacturonan chains. It's interesting that in this case, the cell responds with an increase of extensins. These models are still under study, so this is what we know so far. There's much more to put in that requires further investigation, of course. Then we have Aedes aegypti. Aedes aegypti is responsible for the powdery mildew disease. It affects mainly chlorophyllc tissues, so we can find it in leaves, in green berries, and because it's a biotrophic fungus that requires the living host and the chlorophyllc tissues to survive. So what it does in this case is that he develops an apresorium, so it's a slight tiny leg style that will infiltrate without destroying, and it will feed on the nutrients that are on the cell already. What happens in the plant is that this breach is trying to be sealed with callose plates that are disposed across the breaching point. And last but not least, we have Ascii disease. Ascii disease, it's quite strange in a way that it's a complex of fungus. It's not one species, it's multiple families that are all together in the plant, and they are mainly in the trunk. So they get in the trunk and they destroy it, and at the end of when the circumstances are worse, the plant starts to develop a leaf symptomatic effect. There is the tiger stripe there, as you can see in the picture. But so far there is no report on heavy symptomatic berries with Ascii. However, our studies show that there is indeed changes, and it's mainly in the pectins. So if you can see now in the model compared to the others and the normal model, cell wall in Ascii disease has much less homogalacturonan and rhamnogalacturonan. The rest is still unknown, and we are studying on this. If you have any other questions about the poster, please feel free to come to me or ask any questions, send an email, because we are available and we are more than welcome to share our knowledge with you. Thank you!